You've been here all week, Jasmine. Well, Jasmine was the Jasmine from today. I did some live catching up with some of our paid users in London today. Shout out to the Trampery as well. If you want to know where I am, it's basically I am in a cool, swanky office building in London. And yeah, they're letting me stay late. There's actually, if you hear any little music, that's why I'm using my headphones today, because there's a little bit of, it's a Friday social basically. So yeah, that's exactly what's happening. Hi, everyone. Okay, so what are we going to be doing today? Well, basically, who was here for all the AS biology answers in 1 hour? Who was here for that? Okay, score out of 10 for usefulness for the one you've already done. Mega, okay, so we're going to be doing like as close to, I'm going to rip through as much of the content. Obviously, I'm trying to do in an hour to like refresh. I'm trying to put as many of the answers that are going to be on your exam on the screen in 60 minutes as I can. That's my challenge when I sit there and I plan these. I forgot the link to the paid web class series. Hassan, I don't quite get what you mean there, Hassan. So let me know, can you describe that a bit better? There's only a few, like numbers are trickling up. We're getting, hello, if you've just joined us, who's excited? Because we're going to get through lots of content. Okay, I can see your messages. If you send it to the panelists, it's just me who gets to see it. If you send it panelists and attendees, you guys get to do it. So what we're going to do with the chat is basically the chat is going to be for biology talk only. And the Q&A, you can send questions in there, although I'm going to only be able to check that periodically. There's like mega amounts of volume here tonight. So how many people are here? So we're just before 8 o'clock and we've got well over 50 people in here already. So how many people do I have? I don't quite know. In the web class right now, about 50. I'm expecting anywhere between 300 and 600 to join between tonight. It's a Friday night though, and it's 8 o'clock. So mind you, it's also revision time. So people are not out sinking beers. Revision night party. Yeah, well, somewhat. Can you hear the music in the background? Good. Yeah, I mean, it's kind of chill house electronic. Who uses the Tailored Tutors revision playlist on Spotify? Okay, so the revision playlist, do you like them? This is something we've been trying out this year. Like we've created some that we use in the office basically when we've got heavy work on and we just want to like tune in and zone out to just work. If you haven't used it, if you're big Spotify users, you've got Spotify going on in the background whilst you're revising headphones in, then check out Search Tailored Tutors. You will find it and you will figure out what's going on. I might actually resume the recording. So hello to everyone watching the recording. You should have been here live because you could have asked me some questions, but we are going to get on with things and get some biology movement. I'm not going to start with it. I'm going to start on the Be the Bang with the all intro once we've got everyone online. We are going to start at 8 o'clock. When does it? Classic questions people ask every web class. When does it start? When does it end? Where can you find the recording? Recording, where you just click join this live web class on that button, 24 hours. So tomorrow, this is going to be this recording is going to replace that. And then you'll be able to just watch the recording until the end of the week. Yes, we're starting at 8. We're going to finish absolute hard limit, 9:30. These guys have been super nice to let me stay. And like I'm supposed to be out of here hours ago. So in which case, I promised them I'm walking out the building. So like we're going to do absolutely no later than 90 minutes today. I will do it. As always, I'm going to try and give you as much content as I can during that time. We're basically doing all A2 stuff that's common to OCR, AQA, and EdXL. If it's on only EdXL or only OCR, I'm not going to go into that today because it's just going to get confusing. But all the stuff I'm going through, there's a tiny little bit in photosynthesis where, like, for example, triphosphate, OCR, and AQA, EdXL students call it GALP or JLP. So I'll let you know. Anything that's going to be example specific, I am going to let you know. Hassan, sorry, it's just jumping off my screen so quickly. I did see that message just come through. You wrote it out for me. Yeah, in the paid way. Okay, so yes, absolutely, you're totally right. I had to change the schedule. Basically, in full fairness, I wasn't totally well enough organized to get. I planned a paid web class for today, and this is replacing that. I will be going through practical stuff in the rest of the paid series. So if you guys decide to buy the revision web class series, which you totally should, because it's going to be like this, but better in that there's going to be fewer people in the room. I'm going to get to all your questions a lot more. but I forgot to post the link for the web class. The web class didn't happen. I did forget to edit that class though, so that is my bad. I'll probably send a message to everyone who was enrolled in the revision web class series who paid for those web classes, and I'll make sure that no one's paid for it and they didn't get anything. So but thank you for letting me know, Hassan. That is good to know. It's not the practical skills tonight because I thought this would be more useful. I can add more value to you guys by ripping through, because this is one of the last free biology specific web class. The web classes on tomorrow and Sunday are going to be super useful to you, and I'm going to wait till 8 o'clock so I can actually talk about them properly. The message is flying off my screen. How to win a course. In fact, we're going to start with that at 7:59. I think we can probably get cracking. We've got over 100 people in the room, so we should probably do that. Recording has started, and it's also on YouTube. But I would, if you're here and there, definitely watch this one. This one, like, I ask a question, I get replies straight away. YouTube, there's a huge lag, and it's a bit glitchy. I was looking at trying to do the whole thing through YouTube, and it was just like, just write it off. Okay, all right. I'm going to pump my screen share on. Who's enjoyed this week? Who's like, well, maybe enjoyed is the wrong word. If you actually enjoyed it, you can say, let me know. Who's found this week useful? Oh, we're being joined with a Ronnie. Hey, Ronnie. She's just finished her maths web class. So she's going to be joining us this weekend. Certainly on Sunday, we're going to do a Q&A. Ronnie, Richard, Chemistry Richard, and me. So we're going to announce all the competition winners. I just had a question there. Ronnie is awesome. You bought the all access. Yeah, sorry about the little delay in your all access pass. It was a bit of a technical hitch on my part. Anyone buying the all access pass now, there's just a tiny delay before you get hooked up with your courses.
Oh, look, they're being nice to me, Rich.
Oh, they are. Well, they're always nice to you, Ronnie. Are they not always nice to you? Okay, so we're at 8 o'clock. I can kick off with, like, now that everyone should be here, will be here. And people are always late. I can see that number just going up and up and up and up and up. And I'm going to pump my screen sharing on, and we are going to go through how you can win the course for free, because that seems to be hot on everyone's mind right now. Okay, so those of you who know, how can you win the course for free with Instagram? You've already entered. We're going to draw the winners and announce them all on Sunday. You've got this. It's past paper training week. You see all these comments on there. Tag a friend in there, and you're going to win it for you. You're also going to win it for your friend. So tag an A-level biology friend, tag an A-level maths friend, tag an A-level chemistry friend, tag all of your A-level friends, because every time you tag someone, you're going to get your a virtual name in the hat. And then obviously the more times, the more people you tag, the more they're going to win. But obviously only your friend is going to win it for you, and your friend are going to win it. So basically only tag those people doing those subjects, because otherwise they'll be winning a course but be no use to them. Okay, so everyone got it. Everyone got how to win on Instagram. Currently, there's 193 people. So there's quite long-ish odds of doing that. But if you enter with 10 people, you give yourselves a decent chance. How else can you win? Right now, you got the best odds of winning the course for free right now is to leave a public Facebook review. I haven't counted today, but as of yesterday, there were 7 people who had actually bothered to write like a 1-sentence review. So that gives you, if you're the next person, you're the 8th person, that's a 1 in 8 chance. There's probably a few more than that now. But essentially, like, your highest chance of winning the course this week is to write a 1-sentence review. You're not chosen based on the quality of your review. You're chosen at random. Everyone who's done one this week will be, again, names in the hat. We'll get somebody to pick it at random. Winners will be announced on Sunday at 6 p.m. in that drop-in Q&A session with Ronnie, Rich, and I. The 189 reviews total, but not this week. 189 this week was look at the dates. You can go most recent. Read them whilst you're there. Also, take a second to, like, read the reviews. I don't know. You'll find the date 21 hours ago, but there's, like, I don't know, maybe 20. You're probably going to have, like, maybe a 1 in 50 chance of winning it, which is pretty decent odds for taking. How long is it going to take you? Maybe 1 minute, 30 seconds, 2 minutes. You can write 1 sentence, pick a 5-star rating, click go, make sure it's public. Because if I can't see it, I can't pick you as a winner. We had loads of people last year who just shared it wrote a review that was only visible to their friends and family. No use to me, to be honest with you. And if I can't see it, I literally can't pick you as a winner. Can you do more than one review? One review, unless you've got more than one Facebook account, then no. And it's pretty hard to duplicate Facebook accounts these days. Everyone clear how they know how they can win on Facebook? On your videos, you'll say there's lessons on antibiotics for sure. Okay, all access pass. So who wants to access the course unlimited? Well, between 9 a.m. and 6 p.m. Saturday, 9 a.m. to 6 p.m. Sunday. Would that be useful? You can get your teeth in there. You can see what it's really all about. Okay, well, let's have a look how you do that. Exam training week. So in your, oh, that is not the right place to be. I'm just going to pause my screen sharing just for a second so I can pull up the right thing in here. My internet here is so fast. It's wonderful. Let's pull open the curriculum. I can resume my screen share. Okay, so you guys all have access to this course. That's how you end up in these web classes right now. Okay, so right here, third on the list, how to get a free all access weekend pass. Dum-ba-dum-dum, click on that. Anyone bought any of the courses? And if you have bought the courses, what would be your advice? Do you like it? What do you think? Tell people in the chat. They're going to believe you guys way more than they're going to believe me. I know that it's awesome. Okay, so if you want the weekend passes here, it will give you a little taster. It's going to end at 6 o'clock on Sunday, but don't worry, we're going to follow that up with a web class at 6 p.m. So when you finish that, we're going to be there. We're going to be holding your hand until 7:30. And then when we're finished at 7:30, basically that's going to be the end of this week, but you can then use our courses because our courses are going to be holding your hand, supporting you. Ronnie's going to be there answering comments. If you buy the maths course, you can basically have us as your support team for the next month when it really counts between now and your exams. What about the royal wedding? I live in Canada. I don't even toss about the royal wedding, to be perfectly honest with you. When is the royal wedding? And I don't even know. Is it Saturday or Sunday? Saturday.
Is it this weekend?
Apparently so. Tomorrow. Sats.
How exciting.
What time is it?
More exciting.
So our web class tomorrow is 11 a.m. 11 a.m. until 12:30. They're usually about 90 minutes. So you can see all the schedule here. This is Saturday, 11 a.m. Sunday, 6 p.m. You can make your decision. What's more important, your entire A-levels or the royal wedding? But obviously, you can take a bit of time off to watch the wedding. Of course, the wedding, my life. Well, you can make that decision. That's not for me to decide. That's for you guys to decide. Everyone clear how to access the course all weekend? Any questions, any mysteries, anything that's confusing there? When you fill out this form, basically, we're going to send, all you got to do to join it all weekend is bring a friend. We want as many. We've already had to add another 1,000 coupons because the 1,000 have already gone. So bring more friends. I want to max out the full 2,000, but there will be a hard limit on 2,000. So if you haven't done it yet, make sure you do it because I don't know what the current count is at, but there will probably come a time at some point today or tomorrow where that pass is no longer available. You've got your coupons. Okay, the good question there from Tom. If you win the course, if you buy the course between now and then and you win, it won't affect your chances of winning. We just give you a full refund, of course. We will just refund everything for free. And okay, so there is a special offer going on right now, folks. So I told my class about it way too late last time. Whilst I've got your attention, there's 197 of you looking at me right now. So what the 1/3? Okay, so the deal is, we know it's a lot of money for you guys to buy it on your own. We also know that it's amazing value at the price that it is right now. You're getting some of the best teachers and exam professionals in A-levels in the country to give you hundreds of hours of their time and their coaching in a way that's going to transform your grades. So we know that it's awesome value. However, we also know it's a stretch. So basically the deal is you've got to go and speak to your folks, to your grandparents, to your aunts and your uncles. Go and have a chat with your relatives to say that you've been doing 2 years of work, that you really kind of need it, you need a little bit of extra help to just push you over that boundary. Sometimes 6 marks can be the difference between a D and a C, or between a D and a B sometimes. Like gray boundaries are so tight. So go and have a chat with them. If you can persuade your aunts and uncles and mothers and fathers and brothers and sisters who've got jobs to cover 1/3 of the course, then you need to cover another 1/3 of the course. And then if you can do that, we will give you the course. We will cover the final 1/3. So basically, go and beg, borrow, and steal 1/3 of the course. You have to find a 1/3 of the course yourself. Who thinks it's worth 20 to? Who thinks it's worth 40 quid to get all of me teaching you all of A-level biology? That's £40 from me. It's £40 from you. And it's £40 from the rest of your family. Like 40 quid literally is our tutor. That's less than 1 hour of my time. If I was going to even think about tutoring you on a one-to-one basis right now, you wouldn't get me for an hour. You wouldn't get me, and you can get me to teach the entire specification. So if you think it's worth £40 and you can get £40 from somebody else, then that's the biology course covered. Yeah, so the course, the revision kit, if you're an AS student and you want it for next year, send us an email. There are ways that you can buy the course and have access for next year. That's totally great value. These courses expire on the 1st of July. If you've already bought it and that's an issue, send us an email. We can fix you up with if you need something different. Email is always the best way to get in touch with us, office@tailoredtutors.co.uk. If you want to have a look inside the course, you can do view kit contents and you can have a good poke around. So the biology kit, for example, includes all of this. Everything here is included. So you can look at the AS course and you can see just how many videos are actually included in here. You've got loads and loads and loads and loads and loads of as much as you got. Literally every single topic in the spec, I'm going to sit down and do a 10-minute tutorial with you, and it's all super duper exam focused. AQA, OCR, Edexcel, all covered. And then to actually buy it, it's actually probably not going to let me buy it because I already have it, but we'll see if this is going to work or not. Yeah, okay. So you go to the checkout page. It's going to be 118.80. We call it 120, but it's actually a little bit less than that because the VAT is applied to it. And then it's 1-3 uppercase, lowercase, doesn't matter. Apply the coupon. It takes 33% off, and happy days. Okay, who wants to do some biology? Yeah, everyone know how to get a third off the course. Who's going to go and tell their friends they should go and buy it because it's bloody damn awesome? Already bought it. Awesome. Well, I'm going to stop. Already did. Pass it on, move on. Let's get going, Rich, is what you're saying. Without further ado, this is all the A2 biology answers in an hour. What does this look like? I've just shown you the title. What does this look like? We're going to do respiration first. And again, I'm going to show you, I'm going to give you a demo inside the course. So this is glycolysis. When you have the course, when you get a chance to use it this weekend, you're going to see the video. And this is the best way of revising this. The biochemistry is super technical. There's loads of little details that you need to know. But if you can draw out these, what I call skeleton diagrams, empty box, and you can put all these curly arrows on it and you can just bang these out. You can draw this and then draw it empty, photocopy it, scan it, do whatever you want, and then you print out 10 copies. And then when you've got 10 copies, just sit down, try and fill them in, test yourself. If you don't get them all right, go back, make sure they are correct, and then do them again and again and again and again. You're going to be able to get to the point where you can just do this, basically. Okay, what does respiration start with? What goes in box number 1? Everyone should be able to tell this by now. Glucose. Okay, glucose. How many carbon atoms has glucose got? 6 carbon atoms. Good. I'm not even checking because I'm assuming you're going to know. Okay. The first stage, we've got to charge up glucose. Glucose isn't that reactive. So we've got to amp it up. There's a color coding system. This is going to match the color coding system that I use throughout my course. It's going to help make it more memorable. So when you get to the exam, you can visualize the page and be like, I can remember ATPs were all in red. So this is going to be ATP. I'm going to zoom in a little bit because I'd like to write a little bit bigger than that. Okay. Oh, that's not very, I'm moving my page. Okay, sweet, that'll do. That'll do. Okay, ATP in and ADP coming out. So we're using ATP here. Do we need to write plus PI? Do we need PI? Yes or no? Split opinions. Okay, what are we doing here? We're using ATP, which means we're releasing energy. But the PI is the phosphate that's broken off. So we've got 3 phosphates, triphosphate to diphosphate to 2 phosphates. What's this molecule? What goes in this box? Let's go back to black. This is glucose phosphate. Oh, interesting. Glitched out. Phos. Okay, so do we need to write PI here? Nope. Because this phosphate isn't being released. It's not given off. It's given to glucose. So glucose can become glucose phosphate. How many carbon atoms in glucose phosphate? Have any been gained? Have any been lost? No, still 6. I'm just going to, whilst I'm in blue, I'll give you a hint. The next one's also 6. Okay. And then what's the next one? Well, it's in red. That gives you a bit of a clue. We're going to add some more ATP. And this is kind of counterproductive, right? We're trying to make ATP here, but here so far we've used 2 of them. That's kind of dumb, but all will be revealed. Again, we're adding phosphate. What's the process of adding phosphate called? This is hexose bisphosphate. Okay, so this process of adding phosphate, a useful term to know, is phosphoryl, YL, if I could spell. This is an H, by the way. Oh, let's undo all of that again. Ation process. Phosphorylation. So we're charging up glucose. We're making it more reactive at this point. It's so reactive, in fact, it splits in two. It's oh, I can't, I'm going to have to zoom out because I need to move the screen and I don't want to move my document. There we go. Okay, so the hexose bisphosphate literally snaps in two. It doesn't need an enzyme. Oh, maybe it does, but you don't need to know an enzyme. To be honest, I don't know if it needs an enzyme or not, so you definitely don't need to know. Okay, it now splits into two. So these are exactly the same. I'm only going to fill in one side just to save time. I'm going to do this left-hand side, but what's going on this side is identical to what's going on this side. So again, sketch out this empty flow diagram and then test yourself by filling it in because you need these technical details. That's where the devil is with this respiration questions. We've got 3 carbons and 3 carbons. We've not lost any, we've not gained any. It's just that now we've got 2 of them. It's split in half. Everyone happy with that? What's this molecule called? This is triose. Be careful, especially AQA users. You cannot call TP anymore. They've removed the acronym triose phosphate from the spec, so you must call it triose phosphate. Okay, now what's the final molecule of glycolysis called? Pyruvate. You can also call it pyruvic acid if you want to. Same. Okay, let's go. Let's do what's representing ATPs and ADPs. Okay, here we've got a 2-ADP to 2-ATP. So this is the same on both sides. I'm actually going to add these in. Okay, at this point, how many ATPs did we use in the top section? We used 2. We used 2 up here. How many have we made in the bottom section? We've made 4. What's the net yield of ATP? So 4 minus 2 equals, that's maths that I can do there, Ronnie, is 2. So the net, we've gained 2 ATP. We had to use 2 in the beginning. We made 4 in the end. With the net yield is 4. What's green for? Oh, this green's pretty nice. Okay, so then we get NAD to NADH, reduced NAD, and we get, it's just one of these, but obviously it's happening twice. So in total, how many NADs do we make? In total, we make 2 NADH, or reduced NAD. And what's this phase called? This is oxidation. Redox reaction. So whenever something, so we've reduced NAD, but we've oxidized triose phosphate to make it into pyruvate. Because this literally, so the triose phosphate has a hydrogen atom on it. It gives its hydrogen to NAD, so NAD becomes NADH, which means it has lost a hydrogen. Oil rig, oxidation is loss of hydrogens or electrons. So it's lost hydrogen, so triose phosphate has become oxidized, and in the process, when anything becomes oxidized, the other thing must be being reduced. So you might see the term, if you're a chemist, you'll have heard of redox reactions, because reduction and oxidation, one goes hand in hand. I can't give away, if I give away a hydrogen, yeah, I'm being oxidized. The thing that then takes the hydrogen is being reduced. So it's like go pairs hand in hand. Okay, so down here, these are the things you must know. Where does glycolysis take place? Super classic question. In the cytoplasm, not inside the mitochondria. What's the net yield? We've got 2 ATP, but 2 NADH, to reduced NAD. And where do they enter? They go to the, and be specific here, they want you to be specific, the matrix of the mitochondria by, it's not often they ask, but it is active transport, just so that you know. This is why when they say the net yield of ATP from a complete respiration of glucose, it's a bit iffy. You get various answers because sometimes they include the ones that you need to pump the glucose around and all that kind of stuff. Okay, link reaction. Very few questions on link reaction. I'm going to blast through this. So we start with pyruvate. I'm just continuing from, and how many carbon atoms did pyruvate have? 3 carbon atoms. So we kind of need to know this. It seems like minor details, but we do kind of need to know. Okay, blue is carbon. How many, what's coming out here? We've got CO2. So we've got 1 carbon atom in the form of CO2 being released at this point. So this is a decarboxylation. Whenever carbon dioxide is given off, decarboxylation. So carbon dioxide is removed from the system. So therefore, how many molecules of carbon dioxide, sorry, how many molecules of carbon does this molecule have? 3 minus 1, 2 carbons. Boom. I don't exactly know what. So what's this wiggly line representing? This is so maybe the inner, the cristae, the folded membrane, just demonstrating that this is entering the mitochondrial matrix. And we end up with acetate. And what's green representing? Again, the color scheme is going to really help you. So this is basically we're charging up the batteries. We're always going to be NAD to NADH in the beginning stage, like charging up until we get oxidative phosphorylation. Then we're going to use all those stuff that we've charged up. Okay, oh, it's a green line. I didn't want to put on the screen. Okay, so acetate joins on with something, coenzyme A, and this forms acetyl coenzyme A. How many carbon atoms has it got? We don't know it. We don't need to know how many are in acetyl coenzyme A, but we do need to know that coenzyme A may contain carbons, but they're never going to ask you about how many carbon atoms. I actually don't know whether this contains carbon, but in terms of our principle of chasing our carbons through, you can say CoA. Yeah, that is, I think, is an accepted shorthand. I think this was location that I was looking for here. I don't know exactly what I intended for myself to write there. I'm going to skip over it because it can't be that important. Okay, so location of link reaction. Oh, I want to be in black. Matrix. Important to say matrix. Mitochondrion, no marks. Matrix of the mitochondrion, 1 mark. Products per reaction and per glucose. Well, we split, didn't we, at the top here? So now I'm drawing as if I'm continuing down one of these branches. Yeah. But for everything that I'm drawing, it's going on paralleled elsewhere because we started with 1 molecule of glucose. So basically we can double it for the products. Products are going to be 1 times NADH, and we've got 1 CO2. Yeah, so therefore it's going to be 2 NADH and 2 CO2 if we're per glucose. Something is something and something to form something. Okay, so we can say pyruvate is oxidized, and I'd say is oxidized. Okay, I'm not sure. Let me go back to my lesson because that's definitely what I planned it off. So again, all of these are covered. If I'm going to skip to the end of it, we're going to pause it. Pyruvate is, we could say, dehydrogenated and decarboxylated to form acetate. What do these words mean? Dehydrogenated, removal of, removal of hydrogen, decarboxylated. These are not essential key terms, but they show that you know what we're talking about, and they are important to understand because they do come up from time to time, and they've often thrown students off in the past. It takes place in the matrix. Again, all of this is in those notes. So they're as accurate as they can be. This is what you need to know. It's not coincidence that I've designed the lessons to be that way. So we've done that. Next stage next stage is called Krebs cycle. Again, you should be able to basically just rattle these off because I'm going to go a little bit faster now. Okay, OCR students need to know the names of these 2 compounds. AQA students and EdXL students do not. I'm going to put the names in. So at AQA and EdXL, all you need to know is the number of carbon atoms. This is 2. Oh. Okay. So you need to know it's 4 carbon plus 2 carbon equals 6 carbon. The 2 carbon we need to know the name of. Everyone needs to know the name of. This is acetyl-CoA. Okay, Edexcel and AQA, that's all you need to know. OCR, you need to know. This is oxalo, if I could spell oxaloacetate. You can say oxaloacetic acid if you prefer, it takes longer to say. And this forms citrate. You may, if you're YouTubing this, you may see this called the citric acid cycle because this is citric acid or citrate, basically vitamin C. Okay, blues, carbons, we lose one carbon in the form of CO2, therefore 6 minus 1, we must have 5. 5 minus 1, we must have 4, which we've got over here. Wonderful. Okay, carbon atoms dealt with. Decarboxylation, decarboxylation, whenever we're losing carbon. You might get this is a massive simplification. This is, how do I remember my first year biochemistry exam? 8 reactions, I think, and we look at 4 of them. So there's at least 1 reaction between each of these stages. It's just a massive simplification. We don't need to know the name of this. It's a 5-carbon compound. It's alpha-ketoglutarate, I think. Does anyone know? I think it's alpha-ketoglutarate is the 5-carbon. There's probably a couple of 5 carbons. Yeah, I think it is. 5C. You don't need to know. Okay, green is our coenzymes. And again, you need to, this is going to take a little bit of practice. Draw it out empty, copy it 10 times, and then fill it in 10 times, and you can just rattle these off. This is NAD to NADH. So we're always charging up. We're always making the reduced version of the coenzyme. Always, always, always. And then here, this one is just a bit tricky because there's 2 NADs to 2 N. Oh, that's not an N, that's an A. Right. And NAD. Okay, so what's the other green one? This is FAD. FAD to FADH, reduced FAD. Okay. If you prefer, you can draw NAD twice. We've got 2 NADs. You could draw 2 curly arrows and do NAD, NAD if you want to. You got it. Okay, and this is, again, red ATP. And we make a molecule here, ADP to ATP. What do we call this method of making? We're not doing oxidative phosphorylation here. There's a method. Anytime we're making ATP without oxygen, there's oxygen is not required in here, is called substrate level phosphorylation. That doesn't come up all that often. If they're talking about how ATP is made in Krebs cycle, this will be a mark. Guaranteed, nailed on. They love it. They never talk about it for glycolysis. It still applies to glycolysis. Never seen a question, any exam board on substrate level phosphorylation in glycolysis. It could be, if it said, name the process by which ATP is made in glycolysis. Well, we're making ATP here. We're not using oxygen. It's substrate level, but never happens. Never happens. They only ask it in Krebs. Okay, if you want to know how to do this, full video on Krebs cycle, check it out. Yeah, you can go through here. You just skip to the end. Well, I wouldn't recommend just skipping to the end of the lesson because you're going to need to watch it. I'm going to give you a full explanation. But if we need to know, all the details here are what's going to be the missing in the gaps here. Okay, shall we have a quick question? There's a good question on this. And again, you're not always going to get an exact copy. I wrote this lesson before this. This is a 2017 paper, I think. I wrote this before this came out. Okay, so what can we zoom out? Just a nudge. Okay, question readable. What is the definition of substrate level phosphorylation? Well, phosphorylation, the process of adding phosphate. So we're making ATP. I don't know, Google it. Making ATP without oxygen. It's the energy to form that high energy bond comes from the reactions of the cell. I'm flailing. It's not using oxygen. It's not an oxidative process, which is where the marks in the exam will be. Okay, so it's going to ask us to name K to N. So let's go through them. Okay, well, we've got acetyl-CoA, easy. This is an OCR paper, so they would need to name it. AQA and Edexcel, don't freak out. oxaloacetate, citrate. Okay, here, weird. In my diagram, yeah, we've got carbon and NADs coming out together. How do we know which one is which? It is weird, but we can solve this. Okay, well, this is how many carbon atoms? We've got a carbon atom coming off here. It's actually, I'm assuming, because they're giving us CO2 here, actually, I'm changing my mind. They may actually allow, right, the molecule that is missing from the diagram. Well, we can either say NADH, or we can say C. Does anyone have this paper anywhere and can look up the mark scheme for me and tell me whether it's a hard one or the other? It's an OCR paper. I think it's 2017, but I'm not 100% sure on that. My money would be on this. This has more biological importance because it's going to go to electron transport chain and it's going to be used. Also, they've given us CO2 here, although citrate, as we know from our memory, we've got 4 plus 2 gives us 6. So a carbon dioxide is, maybe it is CO2 then, because we know that citrate is 6 carbon. It's naughty. Yeah, I think actually I've changed my mind. Just by looking at it, we know that citrate is 6 carbon. This we know is 5 carbon. So CO2 must have been given off. It's possible that, I mean, it should be both. Maybe we'll accept both. Definitely it's CO2. Okay, I think my answer would be CO2 here, just because I know it's 6 to 5. I think that's the most solid answer. And you will get bits in the exam where you're not 100% sure and you're going to have to make an educated guess. And then so what's the process of adding phosphate to something? Yeah, I don't know the answer to M dead set. I would hedge my bets on M. It might in the mark schemes accept both. Phosphorylation. Yeah, the process of adding phosphate, phosphorylation. And we could say substrate level phosphorylation would be, I'd put substrate level phosphorylation. You shouldn't lose a mark there. Yeah, for sure. Okay, boom. But can you see how my videos are directly linked to the question? Krebs cycle happens in the matrix of the mitochondria. Absolutely. Yeah. And then we move on to the big one. This is where the whole point of glycolysis, glycolysis does make some ATP. So it's kind of anaerobic. We're going to come on to that in just a second. How are we doing for time? Holy shit, it's 8 hours. Okay, I'm going to go a bit faster. Okay, let's just go. Where are we? This is a mitochondria. Where are we? If we're looking at, we zoom in here. Yeah, we've got the outer membrane here. We've got the inner membrane here, and we've got the intermembrane space. So if we look over here, this is the outer membrane, this is my wiggly inner membrane, and this is the intermembrane space. Okay, so I need to go faster is what I'm saying. Okay, so green reduced coenzyme. So we've got NADH. Yeah, all the stuff we've used, the glycolysis, the Krebs cycle, and link reaction has been building these reduced coenzymes, right? NADH, FADH. These are reduced. Now going to give up their hydrogens. They're going to be oxidized, go back to NAD and FAD. Why? Because they need to be recycled and they need to go back to Krebs cycle and link reaction so they can be reused. They're not just one-use, single-use plastics. These are constantly recycled. Okay, the hydrogen that they give off is a hydrogen atom. A hydrogen atom is 1 proton and 1 electron. The electron enters the internal Christie membrane and goes through the electron transport chain. So that's the electron moving through proteins, electron carrier proteins in the membrane. The hydrogen, then the proton is a hydrogen ion, which is this guy, gets the energy from this electrons pushing through here, is going to pump the protons into the intermembrane space. What does this build up? We get loads of protons in the intermembrane space. This is supposed to be saying H+, by the way. You can't really read it. We get a proton gradient or a concentration gradient, loads of protons in here. Is everyone happy how we got that? Because the FADs and NADs gave up. They got oxidized. They gave up their hydrogen. The hydrogen ion moves across here. The electron goes through here. This then, the hydrogen ions, we've got a nice concentration gradient, and the hydrogen ions diffuse back through this protein. What's this protein called? ATP synthase. This is ATP synthase. This is the A's is the enzyme. Synth means making. ATP is what it's making. So what are these red curly arrows? This is ADP and PI into ATP. We're making ATP and we make shit tons of it. Okay, now we've got the electron is flown, it's done its job, it's gone through the membrane. The electron's job is to flow through the membrane, the electron transport chain, ETC. You can call it the electron transfer chain if you want to. That pumps the protons. That's the job of the electrons. The electrons then drop out the end and we have our E minus. And our protons come along that they've gone through ATP synthase. They've done their job. And big key term. What joins, what links all this up? What's the waste product of respiration other than carbon dioxide? H2O. Okay, so we've got hydrogens. What are we missing? Oxygen. Oxygen sucks up this waste electron. It sucks up this waste hydrogen ion, and we form water. And so we call oxygen the final, you can call it the terminal. I go with final, electron acceptor. Which is why this is called, I can't write. Okay, boom. Done. What is all this stuff? This is like proton gradient. Chemiosmosis is this process of hydrogen ions diffusing through ATP synthase is chemiosmosis or chemiosmosis. It doesn't come up all that often, but it's good to know what that is. Okay, glycolysis we've done so far. We've got two different stages here. In plants, we go TP, and you can't call it TP, but I'm going to write it into pyruvate. Yeah, that's the same as we've done before. In plants, we remove a carbon dioxide and we go to F and Al. That's Al on the end. Yeah, and then this is finally we go from F and Al to F and Ol. This is alcoholic fermentation. Why do we bother doing this? We're losing energy by doing this. We're giving away a carbon dioxide. So how many carbons does ethanol have and ethanol have? Pyruvate has got 3. We lose 1. It must have 2. Okay, so if we've got, why do we do this? Why does the plant do this? Why don't we just leave it as pyruvate? Just stop there. We'll just hold up. We'll just hold on. We'll just store pyruvate because we can use pyruvate later on. We've made our ATPs. We're happy. Pyruvate, it could become toxic in really high concentrations, but why this? What do we need? We need, when we've got no oxygen, we need ATP. This is why we're doing it. This is the magic thing, right? But why ATP generation stops? Because in order to make the ATP, we must also do the NAD to NADH. And we don't have an infinite source of this guy. So we need to reoxidize our NADH into NAD so that NAD can go back and continue to do this so we can make some more ATP. Because if we don't oxidize our NAD, we run out of NAD and then we can't make more ATP. And if we can't make enough ATP and we've got no oxygen, we are dead. So that's alcoholic. It's exactly the same process when we're going from NADH to NAD, yeah, which gets recycled back into NAD. So this can all continue, except what's my product? Here I go from TP to pyruvate to lactate, lactic acid, lactate. Okay, that's enough of it. All the questions on this, 99% are going to be on the recycling of the NADH. Why do you bother converting NADH back into NAD so it can go back and continue the process so we can continue to make ATP? I'm way behind schedule, so things are about to get real pretty quickly here. Okay, this is the chloroplast. I'm going to assume that you can, I'm going to tell you what these bits are. We've got an outer membrane, it's a double membrane, they never ask any questions on it, so let's not worry about that. That's number one. Number two, a stack of these things. A stack is called a granum, grana for plural if we're talking about more than one. One of these stacks is called a thylac, a thylakoid. Why does it have thylakoids? Why do we have, the reason why mitochondria have the cristae, the folded membrane, increase the surface area for the rate of reaction. Which reaction? The light-dependent reaction. Okay, what do we call the equivalent of the cytoplasm? It's called the stroma. Be very careful here because this looks a bit like stomata. It's also a plant word. If there's any ambiguity in your spelling here, they will not give you the mark. This is where the Calvin cycle takes place. Okay, and the things that join them together are called lamellae. And the electrons and a few things flow along them. You don't really need to know. These are lamellae. Lamellae means like layer, basically. Okay, so again, I did the zooming anything before. This is the light-dependent reaction we're about to look at. We're going to look at this bit here. Let's zoom in on this. Yeah, so we've got the stroma on the outside, the top of the thylakoid membrane, and then a bit of the inside of the thylakoid. Okay, happy with that? The zoomy any thingy. It sounds quite nice to say. Okay, so now we're going to move over here. We've got the stroma on the outside and inside the thylakoid here. So can you see now why having loads of thylakoids is going to increase your surface area? Because this is the light-dependent reaction. So what's going to happen? We get some light coming in. It's going to hit the chlorophyll in. This is actually photosystem 2. Rather annoying. They found photosystem 1 first, which is why it got the name 1. But chronologically in the sequence, it's not first. Whilst I've got a yellow out, we need light hitting 3 different things here. Okay, so light is going to hit the chlorophyll in photosystem 1. This is going to excite an electron. So the electron is in chlorophyll, it gets the light energy, a photon hits it, bang, and the electron leaves the chlorophyll. Sometimes AQA will call this photoionization, because if the electron leaves, you make an ion, and the electron has left chlorophyll, so they can call it photoionization. What do we call it when an electron is flowing through electron carrier proteins within a membrane? What's this called? In fact, I don't want to write it there because I'm about to draw something there. This is an electron transport chain. This is why you don't call the final stage of respiration electron transport chain. The final stage of respiration is oxidative phosphorylation, because an electron transport chain is an electron flowing through proteins within a membrane. Okay, this electron, does chlorophyll have an infinite supply of electrons? Does the universe just, no, it doesn't. So it can't just keep giving away electrons without adding more electrons. Where do we get the electrons from? Photolysis. So photo, using energy from light, lysis, to split. What are we splitting? We're splitting water, H2O. So again, we break it down. We have our hydrogen ions and we have our electrons, which I should do in red. And the electrons go into the chlorophyll to replace the ones that have been lost. Oh. Okay, this again, electron transport chain, we've got energy being released here. Basically, we're going to pump some protons. Anything look familiar here? We've got a proton gradient. What's this molecule? Chemiosmosis is the process. This molecule is ATP synthase. So it's the same molecule, same shit, different smell. We've got the protons being built up here. We form a concentration gradient. The protons diffuse through ATP synthase. And we form ATP. Okay, so again, we're taking energy from the light, so we're building up the molecules. This is the charging up phase. What happens to this hydrogen and what happens to this electron? Well, in photosystem 1, we absorb more light energy. The electron gets so much energy, it leaps out of the photosystem entirely. This is a one-way system. This is technically non-cyclic photophosphorylation, but I'm not going to dwell on that too much here. The videos are in the course to explain the difference between cyclic and non-cyclic. Okay, so now we've got an electron, we've got a hydrogen. They're just hanging around, they're loose, but not for much longer. What do they combine with? NADP for photosynthesis. It doesn't stand for photosynthesis, but if ever you're talking about NADP, it's only you're going to come across this in photosynthesis. So P for photosynthesis. So this sucks up the hydrogen and the electron, and we form reduced NADP. NADPH. Why do you not want it? Yeah. Where do these guys go? Where does reduced NADP and ATP go? That's the question you need to answer. They go. Default colors removed. What the? Okay. They go to Calvin cycle or light independent reaction. Okay. So where do we start? What gets added? What do plants build sugars out of? CO2. How many carbon atoms is in one molecule of CO2? One. It's not a trick question. Okay. An enzyme joins carbon onto something. This something has 5 carbon atoms. This is an acceptable shorthand. RUBP. It has to be a lowercase u. Everything else is uppercase. What's the enzyme called? It's called Rubisco. This is ribulose bisphosphate carboxylase is what it stands for. You don't need to know. Call it Rubisco. Okay. This is where Edexcel go a bit weird. Everyone else in the world calls this. No, in fact, I'm wrong. The Edexcel go weird next. This is glycerate 3-phosphate. Good job I remember this. I've not taught this in ages. How many carbon atoms has it got? It has got 3. Good. It's not a trick question. Okay. So 5 plus 1 is 6. We actually get 2 molecules of it, and they both have 3 carbon atoms. Okay. And that doesn't actually change for down here whilst I'm on the blue pen. Okay. ATP's in red. We're going to now use all that ATP that we made during the light-dependent reaction. So this is going to be ATP in, ADP out. Do we need the PI? This is happening twice, remember. So we've got 2 glycerate 3-phosphates going into 2 molecules of triphosphate. That's why there's 2 lines. We use 1 molecule of ATP for each. We don't need to add the PI because the phosphate is being given to the glycerate 3-phosphate. So it becomes... I'm expecting 2 answers here. Edexcel students, what do you call it? Edexcel, GALP, JLP, everyone else TP, just like it always was. Okay, this is why I don't understand why Edexcel have reverted to this, the same molecule, but yeah, this links to respiration way more. And what else do we need? We need NADPH going in. NADP coming out. And then finally, to rejoin some triose back together to make RUBP, we need some more ATP. We're done. I'm not going to go over that details because we are pooping ourselves. If you need more help, buy the course, check it out in the course videos. Muscles. Okay, so again, generally speaking, we've got to know what these things are. Draw it out empty, sketch it 5 times, then fill it in. Well, let's start with, A is a bit of a tricky one. Okay, let's start with D. D. This is okay. What is the thick, and I'm actually going to start somewhere else entirely. The thick protein filament here is myosin. And the thin protein filament is actin. You might get this in a micrograph. You might get this in a diagram like this. Okay, the myosin is thicker, which means when we look at it, it's darker. And so it's DA. You see how it's darker because there's more shit there? It's the A band. If I could write English. Okay, that leaves us with the bit, which is B, where there's no myosin. And it's lighter. It's the I band. Okay, so what's A representing? What's the bit in the A band that doesn't have actin? This is the H zone. What do we call everything between these 2 lines? You are quick tonight. This is a sarcomere. Okay, what do we call the lines what do we call these lines? These are the Z lines. You guys, before I've even said it, that's why I was talking about Anyone was in my web classes all year? Any Ultimate Guide subscribers? Do you prefer GoToWebinar or do you prefer Zoom? Zoom, GoToWebinar, Zoom, GoToWebinar. Zoom's way more responsive. So much faster. Okay, so this is a classic question. What happens to these things when this is a relaxed sarcomere because there's not very much overlap between the actin and the myosin. When it contracts, the actin is going to get pulled in towards there is a line down the middle, which is called the M line. You don't really need to know that. It's almost never examined, but this gets sucked in this way. This gets sucked in this way. The whole thing shortens up. Okay, so what happens to the I band? Get shorter. Classic question. Yeah, because this bit's more or less going to disappear. But the A band is everywhere you've got myosin. The myosin doesn't change length. So what happens to the size of the A band? A band stays the same. Okay, so the letters can help us out. The I band gets shorter, the A band stays the same. I'm not going to do the calculation question. There's not enough time to figure out going through that. Okay, you could also get it looking like this. Yeah, you could get, okay, now we're going to have to figure out what's going on here because it doesn't look exactly like the diagram. A to B, this is our, basically, we've then got our light bits and we've got our M line, we've got our A band. Okay, so let me just tell you what these things are. A to B is a sarcomere. These are our Z lines, therefore. So this is the A band. Yeah, everything where it's darker is the A band. Our I band is everything in here. In fact, let's hash it the other way. This is the I band. And then the H zone, yeah, it's hard to see, actually. They won't ask you for the H zone. It's almost impossible to see on that diagram. What's D? Mitochondrion, exactly. So glycogen granule, it tells you. What's a string of muscle called? What's one of these strands called? A myofibril. You got it. Myofibril. Often they seem to like magnifying it, so know your magnification stuff. That's all of them. Calculate the distance. That can get. You don't want to do that. Okay, why is glycogen granule present? Storage of glucose. Glucose is going to be hydrolyzed. And it's going to, for respiration, to make ATP for muscle contraction. Okay, so glycogen into glucose, glucose via aerobic respiration to make ATP for muscle contraction. Obviously, muscle contraction, highly energy consumption. During vigorous exercise, the pH of skeletal muscle tissue falls. This fall in pH leads to a reduction in the ability of calcium ions to stimulate muscle contraction. Suggest how. People are telling me they hate suggestion questions, so I did my work to try and put some more in. Okay, so this is saying, let's decode this question. 3 marks, suggest is a command word. We need to get the question clear in our head. We need to say, basically, it reduces the ability of calcium ions to stimulate muscle contraction. So this is saying, what do calcium ions do during muscle contraction? And what would be the result of reducing their impact? Okay, so if you're at Excel here, you need to know about troponin. If you're not at Excel, you can basically forget troponin and go straight to tropomyosin. So the roles that this is classic. What does calcium ions do is a classic question here. They love it. So all the exam boards, this is not just any of the particular exam boards. In fact, look at this question down here by some coincidence. Whilst I'm on it, I'm just going to let the question answer it because we don't have time. If I put that, it's going to be really hard to read. So describe the role of ATP and calcium ions and bring them out of contraction of myofibril. So this question is like basically the same, but saying if there's less calcium, what happens? For which we need to know what happens. Okay, so calcium, basically, it stimulates anyone know the name of the enzyme that breaks down ATP, that splits up ATP into ADP? The only ever comes up in the spec here. It's ATPase, not synthase. ATP synthase makes ATP. ATPase is going to split ATP. It's not ATP synthase, it's ATPase, okay? And calcium is going to make ATPase more active. And so the calcium ions are going to promote the hydrolysis or breakdown of ATP because they're going to make ATPase more active. This provides the energy. What does ATP do? ATP provides the energy Okay, so we've got the binding site, actin-myosin. Actin-myosin binding site. It's like Velcro. Does sticking Velcro together take any energy? Velcro is quite happy to stick to each other, right? You can call it ATP hydrolase. Same word. Same. Okay, so sticking Velcro together, energy free. But now we're going to, once we've got the binding site, we're now going to ratchet across. We're going to pull that actin. That actin is going to be pulled into the myosin. So that ratchet stroke, that power stroke requires ATP. Yeah. Now we've got Velcro, ripping Velcro apart. Does that take energy? Yeah. So breaking that cross bridge is going to take energy. And now I've got to take my arm. I've got to move my arm back to the starting position. This also requires energy. So the things that require building the cross bridge, in my opinion, does not require ATP, although sometimes the mark schemes do give you a mark for it. The power stroke, pulling that, moving in, breaking the cross bridge and resetting the myosin head back to the original starting position, they all require ATP. Calcium ions are going to bind to the troponin. So let's go back and answer this question a little bit more with a little bit better. Okay, so if the ability of calcium ions to stimulate muscle contraction decreases, so what does calcium ions do? So we can say ATPase will break down less ATP, so less ATP available for muscle contraction. We can say that less calcium will bind to the troponin if we're EdXL, tropomyosin if we're everyone else, and therefore it will not change shape, and therefore fewer binding sites will be available. Therefore, less cross bridges will be formed, and therefore it will result in a weaker muscle contraction. How does the pH fall? You don't need to know how the pH fall does it. This question doesn't require any information for telling us an increase in hydrogen ions will reduce, it says reduce the ability of calcium ions to stimulate muscle contraction. So it's just saying that you don't need to know the details. You just need to know what happens, what does calcium ions do, and how will having fewer of them affect the muscle contraction. Okay, and if we want, describe and explain the role of calcium ions in ATP and muscle contraction. The sarcoplasmic reticulum is where the calcium ions are stored. This is an old spec question. This is suitable for EdXL now. You don't need to say this if you're AQA or OCR. Binds to troponin. Tropomyosin moves from the binding site. So myosin head attaches. ATP is needed to remove the calcium ions. ATP provides energy for changing the shape of the myosin head. ATP is required to break the cross bridge. ATP for synthesis of neurotransmitter. Okay, that's beside the point. It's not really that relevant. Whoop. So that's how a development of the leak. Do I need to look at this? Successful development of a leak in a specific class of calcium ion channels in muscle cells can lead to muscle fatigue. Okay, so this is the same question, different wrapping. If there's less calcium, how are things affected? Calcium ions leak out of the cell. There's a change in there's fewer calcium ions bind to troponin. So there's less binding sites available. So less binding sites exposed. Therefore, the fewer cross bridges are formed. Yada, yada, yada. I think I've kind of gone through that. Everyone happy with muscles? I think let's move on. Okay, I'm getting yeses. They're the answers to before. Okay, so the answer to this is actually here. In fact, interestingly here, they did actually, how does it? I said you didn't need to know. Well, we can say that the pH, what is a calcium receptor? It's a protein. So what does pH change do to a protein? All receptors are proteins. Proteins do everything, basically. So if the pH changes, the protein is going to have its internal bonding affected slightly. Yeah, it does not necessarily going to denature. Before it denatures, it's going to get a bit contorted because the hydrogen ions are going to mess around with the internal bonding, especially the hydrogen bonds. It's going to change shape, and therefore it'll be less effective. But what's the answer that I gave you? I started at 0.2, which wasn't ideal, but can we still get 3 marks? 100%. 1, 2, 3, 4, 5. One of my long lost friends from university has just walked in the door. It's very distracting to not look through that glass wall right there and say hello to my friend. Okay. Got nerves. We've got 20 minutes max. Speciation, gene technology, and the nitrogen cycle. Them's your picks. Speciation is really quick. I can actually go through that really quickly. Gene technology and speciation. It is then, I'm going to do speciation first because I love it, because species, like evolution, is the best thing ever. These are 3 examples from 3 different exam boards. Just going to give you the highlights. So most typically, the questions have been in the past, allopatric speciation. What's the difference between allopatric speciation and sympatric speciation? Okay, we're going to do speciation. We're then going to do gene technology, and if there's time, we'll do nitrogen cycle. I have to go. They're going to kick me out of here at 9:30. I cannot stay any longer, so we'll do Q&A at 20 past at the latest. Okay, allopatric speciation. You've got populations separated by geographical boundary. Maybe you've got a mountain range, and you've got one population here, population A, and population B. They literally gene flow or animal flow. They can't shag each other. Basically, there's no interbreeding. There's no, because they're separated. One could be on an island. One could be between a mountain range. Okay, so allopatric, sympatric. So if we're like I think it's like synchronous. Sim just like is, I don't know what's the Latin derivation of sim. I don't know, but it is basically together. Symbiotic, they're working together, right? So it could be the same. So that they're in the same place at the same time, but they choose not to interbreed for usually a behavioral reason. Could be morphological, although for me, morphological doesn't explain anything, because if they're the same species, they are morphologically the same to begin with. They diverge to become morphologically different. So I don't really think morphology should be an accepted answer, but they tend to give it in the mark schemes. Okay, so this is I think an OCR one. What is the process speciation? This is allopatric. So they're isolated. The populations are separated. A detail of the isolating mechanism, e.g. that they're on other side, they're far away from each other. There's a mutation. Okay, I'm going to give you my verbal answer first because these answers are confusing. So like the classic speciation answers, allopatric speciation. Geographically isolated. There's no interbreeding between populations. That means there is variation in the genomes of these populations due to mutations. There's different environments in the 2 new environments, in the 2 different locations. And that means there's different selective pressures. So each become adapted to their own little area. That means there's differential reproductive success. Those that are more suited to their environment are more likely to survive and reproduce. That's what I mean by differential reproductive success. A selection pressure means that if being tall is really advantageous, more of the tall alleles will be passed on to the next generation. If being short is really advantageous, more of the short alleles will be passed on to the next generation. So differential reproductive success causing a change in allele frequency over many generations, and then you end up with populations that are so different they're unable to interbreed to produce fertile offspring. Okay, so that's the off the top of my head one. Like basically that's where you want to be at, so you can go speciation, isolation, mutation exists, or there's genetic variation in the 2 populations, natural selection, or a description of natural selection, or you can say there's different selection pressures because there's different environments. So one side of the mountains is wet, one side of the mountain, let's go with hot and dry. One side of the mountains is hot, the other side of the mountains is cold, and therefore there's different selection pressures. Some are going to become better adapted to the cold, some are going to get more hair or grow more hair, some are going to be more sweaty. This is an Excel version. Geographical isolation, no interbreeding between populations. Variation exists in those populations due to mutation. It's actually an AQA one. I changed my mind. Different environmental conditions, different selection pressures, different features are selected for. There's different reproductive success. Leads to a change in allele frequency over many generations or a long period of time. An Excel question, geographical isolation, you get the idea. Same answer, different exam board, they're all the bloody same. Okay, sympatric speciation. I'm going to move it over here. I need to move the chat. Okay, so Lord Howe Island is big enough to read, probably not quite. Lord Howe Island in the Tasman Sea possesses 2 species of palm tree which have arisen by sympatric speciation. Tells you it's sympatric. The two species have diverged from each other after the island was formed 6.5 million years ago. The flowering times of the two species are different. Okay, bing, 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 bing. Flowering times of the species are different. Using this information suggests how these two species of palm trees arose by sympatric speciation. So they're on the same island. So it could be that they, at different seasons potentially, occurs in the same place. Okay, if you've got to just hit the nail on the head, even though it's obvious to someone who understands sympatric speciation, give this to a university professor, they would never think to say this because they know it's happening in the same place. That's what sympatric means. But you've got to beat the hammer, you've got to beat the drum, let them know mutations at cause different flowering times. So a mutation has caused a change. It's not caused by the environment, it's caused by a mutation. Then you get reproductive isolation. Now the answer is basically exactly the same. It's just a different starting point. Reproductive isolation, so not geographical isolation, but they are reproductively isolated. There's no interbreeding, but no gene flow, no interbreeding. Different alleles are passed on or selected, or you can say there's a change in allele frequency. You could say disruptive selection. Don't worry about disruptive selection too much here. That's an AQA only thing. Eventually, the species cannot interbreed to produce fertile offspring. Make more sense? Understand the difference between sympatric and 9 o'clock. I've got to go. I've got to move. Okay, gene technology in 10 minutes. How are we going to do this? Okay, so you need to know, this is the best summary question. Basically, how do you cut out genes? How do we get a gene on its own? There's 3 ways. I might just go freestyle off this and just tell you what you need to know. So how do you isolate a gene? Well, there's a couple of different ways. We can go to the source. We can go to the DNA itself. In fact, I'm going to draw my DNA horizontally. We know that the target gene is in here. Again, the course is an awesome explanation of this, even if I do say so myself. This is our target gene in the DNA. We're going to use a restriction enzyme to cut at these sites. So anywhere out, so as long as we can't cut inside the gene because we'll mash up the gene and break it. So we need to find somewhere on one side on the other, and this is a restriction enzyme. Or you can call them restriction endonuclease if you want. Restriction enzyme. You can't read my writing, but you can hear my words. Okay, so this is going to cut at a palindromic site. A palindromic site is, you can call them a restriction site, you can call them a something else site. But yeah, restriction sites are fine, and they cut. So we're basically going to leave. Oh, what's going to happen if I use my rubber on this? Yeah, it's going to take it all away. It doesn't matter. I can put it back again. It's going to leave sticky ends. Unpaired bases. Super duper important in gene technology because you need those. So now we've cut it out, and now we've got unpaired bases here. Dum, 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 dum. Dum, 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 dum. Okay, these unpaired bases, if we were to use that same restriction enzyme to cut open a plasmid, for example, plasmids are double-stranded little circular bits of DNA from bacteria. I've done my colors backwards. I'm an idiot. Oh, how good are those circles? Pretty pleased with them. Yeah. So if we use our restriction enzyme to chop this open here, if it's the same enzyme, these unpaired bases are going to be complementary to these guys. And these unpaired bases are going to be complementary to these guys. So they're going to line up. They're going to have, but at this point, they're still, what's the bond between 2 strands of DNA called? Hydrogen bonds. Hydrogen bonds are not strong enough to hold this in place. We need to reform those phosphodiester bonds to reseal the DNA to make it whole again. So what we're going to do, we're going to open this guy up. See if I can do where's my overlap? My overlap is here. Okay, so this is my plasmid. I've just opened it up, and now I'm going to insert I'm going to just color my sticky ends a funny color right now. I'm going to make them green. This bit here and this bit here is going to be this bit here and this bit here. My target gene is still in the middle, untouched, and then the rest of the DNA doesn't really matter. It's just going to be the gap filler. So we've got a gap here. This is all continuous. They've got a gap here. Yeah, I've got sticky ends on both of them. It's not very clear, is it? Let's do that again. We've got this bit is attached to here, and that's cut off. That's the sticky end belonging to here. This is the no sticky end, this bit. And the sticky end belongs to this piece of DNA. So the gap, there's a gap here, and there's a gap here, and there's a gap here, and there's a gap here. We need an enzyme to seal these guys up, to reform these phosphodester bonds, to zip up. In fact, zipping is the wrong term. This is DNA ligase. Okay, so that's the enzyme we're going to use. So this is how we chop the gene. What's the problem with cutting out the gene at the DNA? There's a bit of it, like how many copies of each gene do you have? Each allele do you have? You might have 2 if you're homozygous. You might have 1 if you're heterozygous. So, and where is it? It's in the nucleus, folded up around those histones, like totally coiled up, supercoil inside the nucleus, right in the middle of the cell, super hard to get to, and you might only have 1 copy. That's a bit of a, that's difficult. So let's say this is the gene to make insulin. We want to copy the insulin gene. Okay, where else could we go? So we've got the insulin gene. How many copies of the insulin protein do we have? Loads of it. So this cell, if it can make insulin, if it's a pancreatic cell making, so we've got loads of mRNA being produced. So if we've got the mRNA from the insulin gene, we've got our mRNA like this, and there's an enzyme that we can actually use to read the messenger RNA to make DNA. So it's doing transcription backwards. Transcription, remember, is DNA to messenger RNA. If we want to do that backwards, we can do this, and we can make DNA from it. We can basically go backwards, so reverse transcriptase. It's transcription backwards, and that's what this enzyme does. And we form a single-stranded piece of DNA, and it's called complementary little c DNA. It's missing a few things, though. If we think about our original gene, if we chop it out the DNA, we've got introns and exons in here. Remember, when we do transcription, we remove the introns, the exons are exported, the introns are taken away. That's already happened. The introns are gone. There are no introns in messenger RNA. So when we go from messenger RNA to complementary DNA, you still don't have any introns. You can't make them up. They weren't there in the first place. So this is OCR. Don't go into this level of detail. I should probably have said that. OCR, there is a question where it is useful, but it's basic. Gene technology. Can't move around the screen. This is an OCR question. How do you get a gene? Basically, a renin can be genetically modified. Outline the processes by which bacteria can be genetically modified to produce renin. Okay, so this is a whole question. The first bit, you can get marks here. You don't have to say all this stuff in an OCR exam to get the marks. Basically, section 2, OCR do way more stuff on vectors. Okay, so there's different ways we can say but this question is basically describe how you can synthesize a gene using any gene technology method. So this answer is like the magic bullet when it comes to gene technology. We can use a restriction enzyme to cut out the gene. We can use a gene probe to identify it. Forget gene probes here, they're not that relevant. Or you can get the messenger RNA and use reverse transcriptase to make complementary DNA. These are all different ways. Or you can use a gene machine, or you can sequence the protein amino acid by amino acid, know which DNA bases, code for those amino acids, and then literally synthesize the gene like a gene machine, or like just building it artificially, synthetically in the lab. Then you're going to add the gene that you've isolated to a vector. What does a vector do? The vector is going to be, it's not a mathematical vector. It's going to be transporting the DNA from one place to another. So typically, they're going to be talking about plasmids. Why? Because plasmids are way more biologically interesting. A phage, what is a phage? A bacteriophage is a virus that infects bacteria. The reason they don't ask about these very often because they do, this is their natural life cycle. A virus, it inserts its DNA, the DNA goes into the nucleus, it then hijacks the cell's DNA to start making proteins. It does everything. There's no questions to be asked on this, apart from how does it, basically there's no questions they can ask. So that's why they don't really ask questions. They ask about plasmids. You need to use the same restriction enzyme so it had the same sticky ends. Use DNA ligase to rejoin the sugar phosphate backbone. And then what do we call DNA that's got DNA from more than one source? DNA from more than one source is recombinant DNA, a recombination. You could take Ronnie's eye color gene and stick it in one of my cells. It doesn't have to be a different species. It can be the same species, but from two different sources, it becomes recombinant DNA. And then how do you get them into a bacteria? You take your plasmid, your recombinant plasmid or recombinant vector, and you mix it, you put it in with the bacteria. The conditions are rarely, so you can give them an electroshock, electroportation, basically freak them out a little bit. Bacteria will suck up plasmids when they're, or calcium chloride and heat shock is the other way. So either electroshock or calcium chloride and heat shock. But basically when the bacteria is super stressed, it can go, maybe this plasmid with these random lucky dip lottery genes on it might help me because I'm in dire straits right now. I am likely to die. And therefore, I will absorb this plasmid with random DNA in it. And I will try expressing those genes because maybe that DNA will save my life. That's why if you really stress them out, they're more likely to absorb a foreign plasmid. Is this the best questions to be doing? This is an Edexcel question, I think, by the look of the type. Well, you've got the question there. I'll pause it on the screen so you can watch the recording and pause it and look at the questions. I suggest you cover up the answers. I didn't actually cover up the answers, which I should have done. Bad me. Gene probes. Marker genes. Why would you use a marker gene? Why, when you're putting in your renin gene or your insulin gene, why do you need to use a marker gene as well? Because you're putting, basically you put like in a soup pot, you put your recombinant plasmids in with your bacteria and you want them to go into the bacteria. It's a very low percentage. Let's say less than 1%. I don't know the numbers. You don't need to know the numbers. But now you've got, like, let's say one bacteria in 100 has taken up your gene. What types of marker genes can we use? There's two major types. We've got fluorescence, so we can grow them on an agar plate, shine a UV light on it. If they've got the marker gene, then they'll fluoresce. If they've got the marker gene, they've also got something else. They've also got the gene that you want, the target gene. So it just allows you to clearly identify which ones have successfully been, what do we call an organism that contains recombinant DNA? Transformed or transgenic. Okay, sweet. You can also use antibiotics because you can then, if the marker gene is antibiotic resistance, you can grow them in that antibiotic, and only those that have got the resistance will be able to will survive, and therefore they're the resistant. They must also have the gene that you're interested in. Yeah, there are some other methods, but you don't need to know in general because that's what nearly all the questions are on. Why are you not letting me control Z?
Everyone, it is nearly 20 past and there's 23 questions in the Q&A for Rick.
Okay, let's jump over to there. The nitrogen cycle. My nitrogen cycle on YouTube has been viewed something like 80,000 times. It's totally killer. I can't remember if it's the AQA version or the OCR version, but you're gonna, if you share your friend the access with the course, you will get the all access pass for the weekend, so you can check it out and answer these questions. What I'm going to do, Ronnie, just before we go, is I'm just going to put these questions on the screen so you can pause them during the recording and get the benefit from the answers because I'm a nice guy. Oh, he said, okay, so let's go over. Zoom chat, go away for a second. I need to focus here. I've done that one. Okay, let's just go over here. Let's put all this on the screen. I don't think you're going to. Okay. Jellyfish, jellyfish, jellyfish. Boom, boom, boom. Okay, you might need to see actually that looking like that, because that's probably going to relate to one of these questions. That's enough time to pause it. Gene proby type stuff over here. Again, I've selected all these questions because they're like the classic. I'm not expecting you to be answering any of these right now. I'm going way too quickly for me to even answer them. I'm not even reading them. I'm just putting them on the screen so you can pause it. You probably need the diagram there. I think the diagram lives in this gap. That's really crappy, but I can probably put that on there. And nitrogen cycle. Where did nitrogen cycle go? Whoa. Hey, you folks on YouTube, make sure you tune in on Saturday and do it via the course so you can actually ask, and I can answer your questions. I can see your answers. Okay, it's probably not quite big enough. It's a pretty nasty one. It's an old one, but I'd say a good one. It's not a good one, but it's a nasty one. Done. Okay. Over to me. Q&A. Let's pull that open.
You should have 26.
Okay, I'm going to just request no one sends a Q&A right now. There's no chance I'm going to finish all these, and like adding more questions just means it moves my screen, and then I can't read the question, and then we're going to get less questions answered, fewer questions answered. If I can see the chat, so whilst that's going on, you can send stuff to the chat, and I might be able to pick up your question there. So you do have questions, stick it in the chat. Don't stick it in the Q&A because it's going to make my life a pain. Okay, when is the practical web class rescheduled to? Don't know. Right now, we'll make it public. I don't have an answer, so I can't tell you. Can you write hexose phosphate instead of glucose phosphate? Technically, it is glucose phosphate. Check your marks. Find it in a mark scheme of your exam board. That's not all the same for all the exam boards. I would generally go with glucose phosphate and hexose bisphosphate. But they are different molecules, and so you should treat them differently ideally. Yesterday was meant to be maths, practice, and stats for biology. It was supposed to be at 6 o'clock, but I had to rearrange things. So I will reschedule it and I will redo it. I will let you know when. That's question 4C 2017. Yeah, that's gone. How much time should you spend on synoptic essay? I'm going to do a full web class on the essay. I'd say probably a 10-minute plan, because I'd do 5 minutes. I'd open the exam paper, read the essay question, and spend 5 minutes planning it. Then I'd then go back and do the questions, because you might get some of those questions might spur your imagination, so you can go back and answer it. And then I'd do another 5 minutes on the plan, and then I'd write my essay. If you plan it well, the essay will write itself. Whoa, I've lost my Q&A. There it is. Hey Zara, in glycolysis, do you have to mention hexose bisphosphate, or can you say? It depends. If you need to know the detail, because they can ask like simple 1-mark questions on it. Generally speaking, they're going to gloss over the phosphorylation stage. So they're just going to go glucose to hexose bisphosphate. You probably don't need to know. So generally no, but it is worth knowing, because it can come up. Which topics in A2 are generally harder in the examiner's report? The gene technology is typically done pretty badly. The biochemistry and photosynthesis, if you can do those flow diagrams I went through today, you will get good marks on it. Photosynthesis questions are not difficult, but they are technically, you need to know the details, otherwise you get lost. So do those empty skeleton diagrams and fill them in. I'm not going to do predictions right now. I'll do that in my paid web classes. Photophosphorylation, when does the process photophosphorylation occur? Photophosphorylation is taking place anytime when there's light available. In which stage of photosynthesis and which are the chloroplasts? So it's taking place on the thylakoid membrane, of the grana. That's why you have so many thylakoids stacked into grana, so you increase the surface area. Yeah, seriously, don't add any more Q&As because it just leaves my screen. Did that answer your question? Statistical test, yeah, if you buy the course, the maths and stats course covers the entire, all the stats, all that kind of stuff. When is the practical skills web class? Keep up to date, open our emails, follow, check out the website, and I will repeat it. It'll be a paid web class for those of you who are paid subscribers. Are you allowed? So what abbreviations are you allowed to use? Look at your specification. It will say deoxyribonucleic acid, open bracket, DNA, close bracket. You're allowed to use DNA. If the abbreviation is not on your specification, you cannot use it in the exam. No, you generally do not get marked down for using the plural or the singular. You can say grana or granum. If it's clearly looking for you to be specific and say granum instead of grana, or mitochondrion instead of mitochondria, it will be quite obvious in the question. But no, they do not typically mark you down for being single plural. That's why I don't bother. Is it useful to use the old spec papers for the other exam boards? No, I would say old spec questions for other exam boards don't bother with. There's enough old spec papers for your exam board to use. They are definitely useful. Use the new spec papers for the other exam boards to do the maths and the stats and the practicals that overlap. Sometimes the practicals overlap. Enzyme catalyzed reaction, potometer, some of them don't overlap. So dissection, for example, doesn't, AQA don't do that. Does the past paper scheme include solution examples? Not this year, sadly. Hey, Robbie, was that you today? So yes, basically the all access pass with the third off, the 1/3 offer is £200. If you buy the all access pass, you get all the biology, all the chemistry, all the maths, and all the web classes, all the biology web classes, the paid for ones, all the maths web classes, the paid for ones, and all the chemistry web classes. So you're getting the web classes free, and for £200, you're getting basically everything until the end of exams this year, which is amazing value. Does the process of phosphorylation occur in both respiration and photosynthesis? Phosphorylation, yes, because you're adding phosphate. So oxidative phosphorylation, photophosphorylation. The thing that generates the energy is the light. In photophosphorylation, the oxygen and the chemicals oxidate. If you're basically, respiration is like burning, yeah? You set fire to something, you're oxidizing it, you're releasing heat, and all that, those electrons flow through same stuff. But photophosphorylation is obviously photosynthesis. Oxidative phosphorylation takes place in both plants and animals, but it's a respiration process. EdXLB, I don't know. Sarcomere, sarco. So sarco means related to muscle. Anything with sarco in it relates to the muscle. Sarcolemma, sarcoplasmic reticulum, sarcoplasm. So they're all muscle stuff. Sarcoplasm is the cytoplasm in a muscle cell. Sarcomere is the unit between the Z lines. The sarcoplasmic reticulum is the endoplasmic reticulum within a muscle cell. It's just muscle special terminology. Troponin is in the EdXLA spec. EdXLB, I'm not familiar enough with the EdXLB spec to precisely know. I don't understand, Robbie. So you only get access to them tomorrow? I don't understand what you mean. The all access pass for the weekend is just for the weekend. If you buy the all access pass, you get all access of everything until the 1st of July. So yep.
He bought it, but it's saying that he only has access from.
It's because yeah. I'll fix you up as soon as we're finished.
I've taken his email.
Okay, yeah, perfect. Right now, because the all access pass is going to be, those courses are going to be added at 9 a.m. on Saturday. I've had to do a bit of a workaround. So it's not automatic you get added to the courses. You will get added to them as soon as I finish this web class. DNA fingerprinting. Check out the question packs by topic in the course. The question packs on top of there's loads of stuff on DNA fingerprinting. Basically, it's electrophoresis and PCR under a different name, just both stages linked together. Yeah, this access this weekend you're gonna get the chemistry stuff too, 100%, and the maths. If you're going to start revision now for AS and A2, how would you do it? I'd do past papers and find out where your weak topics are, and then use the videos, buy the course, and you can cram. You could complete all of AS and A2 in a month. It would be a stretch. We've had students do the AS course in a week and the A2 course in a week, so you could do it. You've obviously got other subjects to revise, so that's going to be a real stretch. You can smash through all the videos, take notes, and then do past paper questions to supplement it. I would do both the revision videos and do past papers. Use the past papers to find your weak spots, and then use the videos to fill in those gaps to relearn the bits you don't know. They're all the maths biology specs require the same 10% of the marks are on math skills, level 2 GCSE or above. All of them are 10%. Model answers for topics. Yeah, check out the question packs by topic, all included with the paid for course. Most likely topics to come up. All the stuff that I've done in these web classes are likely to come up. So basically everything in all the answers. Will the webinar recordings be available after the weekend? To anyone who buys the course, yes. To anyone who hasn't bought the course, probably not. I haven't decided yet. The web class, yeah, this weekend you get access. The recordings, Richard is going to do the chemistry web class recordings today or tomorrow. They'll be there before the weekend. Cheat sheet, I'm going to do on the weekend. I don't have any time. It's 9:30 at night on a Friday. I need to go. The cheat sheet will be given out. I'll let you know how to get the cheat sheet when you join the web class on Saturday. The free web classes will be available until Sunday night for sure. Am I doing any more free web classes after this? More than likely not. Possibly, but more than likely not. Access the documents I used today. You're going to have to watch the recording, pause the screen. I got you, Robbie. You're going to be covered in 10 minutes time. Can you just buy the web classes? Yes, they're £29 per subject, which is basically the cost of one tuition lesson. So if you've just bought the all access pass in the last 3 hours, I'll check you out right now and I'll add you to them in a minute. Because my system, I didn't think through my system carefully enough. Okay. At what stage of respiration did heat get reproduced? It's nearly all in oxidative phosphorylation. Especially, yeah, basically it's oxidative phosphorylation. I've never seen a question on it though. Triplets and codons are not interchangeable. DNA triplet codes for the mRNA codon, which is complementary to the tRNA anticodon. DNA triplet, mRNA codon. Thank you, thank you, thank you. You're all wonderful. See you tomorrow at 11 a.m. I don't know where I'm going to be. I might come back here if they're nice enough to let me go, but if I overstay my welcome, they're not going to let me. See you later. Check out the course. Do some past papers. And bloody well go and buy it because I promise you, you will not regret it. See you all again very soon. 11 a.m. tomorrow, 6 p.m. Sunday. Share the unlimited pass with all your friends. Get as many people in there as possible because they will literally, we've had people use the course for 2 days and say that it improved their grade. So see you later.